Thank you for joining us on Research and Science Diaries. I am your host, Fune Cyprien. Today, we are joined by Professor Spitz, who is a professor at the Vrije University, Brussels. She is also the group leader of Genomic Regulators of Cell Fate, which is part of the Genetics, Reproduction and Development Research Group at the Vrije University, Brussels. Professor Spitz, thank you for joining us on Research and Science Diaries, and welcome.
Thank you. It's a pleasure.
Yeah, would you like to introduce yourself by giving us your academic and professional background?
All right, so I am a biologist. I studied in Barcelona many years ago and started a PhD there that I then stopped to move to the Vrije Universiteit Brussel to pursue a PhD in the same topic. At the time, I was working on the genetic testing on human embryos, so PGT, pre-implantation genetic testing. And then when I completed my PhD, they just started the stem cell lab at the university, and they asked me if I wanted to join as a postdoc. And I thought it was, an interesting adventure. So that's how my journey into stem cell biology started. And since then, I have been working on stem cell based models, and still having a bit of a link to the developmental biology and the reproductive medicine.
Yeah, so that's quite a background in genetics. Maybe we would like to know why this interest in genetics and especially in reproduction and fertility.
I think genetics, it's been an interest that grew already while I was studying biology. I think that this idea that there was a blueprint for life inside of all of our cells, and that all of our cells were the same from a genetic point of view, but not from a phenotypic point of view, I found extremely interesting. And I think that was a bit the step into developmental biology, like how do cells actually decide to become what they become while actually all carrying the same DNA. And I think that that's maybe a bit the way I got into this field.
Yeah, we would like to know because with fertility, it's known that fertility it's gradually like maybe reducing across the globe. Could you maybe give us a bit of an idea why do we have a decline in fertility across the globe, or is it an issue of maybe people don't want to have children, or it's actually like a public health concern?
Yeah, I think it's a very complex question, right? It's a multi-factor thing. As far as I understand, I'm not a specialist specifically in the epidemiology of fertility, but as far as I understand, there's different things. There is the fact that women postpone childbearing times until later because we develop a career, and so in fact, the older we get, the less fertile we are, and there is I think a general lack of information on this. I want to say we get like at school sex ed on how not to have children, but we get very few information until what is actually the real time span of fertility, for instance. So we are very poorly informed until when we can postpone the child having children. Then so this is playing an important role. I think the postponing of the time when we have children. Then you have the decline in sperm quality in men. This has been a trend that has been going on for a long time. There's a lot of speculation about this, life factors. So things like dietary problems, maybe all of these endocrine disruptors that are used in plastics, in makeup, in cosmetics. So there's a lot of environmental things that seem to be affecting the sperm quality. So I think those are the two roles, the major roles and the major factors in this decline in fertility. But I am not a specialist in this field. So.
Yeah, you've done quite some research work into artificial reproduction technologies. This technology has been around for over 5 decades. What are some of the key challenges that we still face in this field? And in your opinion, why do these challenges still persist till date?
So from a medical point of view, I think the main objective of having a fertility treatment is going home with a live baby, right? With a baby,
Yeah.
with a healthy singleton, preferably, because multiple pregnancies have complications. And the success rate at this is still not as high as they would like. So there's still room for improvement there. It's a treatment that works really well when you're young, but then again, your fertility is not so diminished when you're young. So there's still a lot of room for improvement and for understanding why some couples don't become pregnant. And so there's a whole genetic basis probably to infertility as well. Not for everybody, but for some couples who really struggle at getting pregnant. So there's, I think, a lot of room still for improvement on these methods. And then there is a whole aspect of safety concerns. And that's what my most recent work has been about. So this is known for a long time that children born after fertility treatments, they have just mild adverse perinatal outcomes. They are just slightly lighter, so smaller birth weights, lower birth weights, smaller babies, a slight increase in like congenital malformations and such. And so this has been still, it's a source of concern of like, why is this happening? Where is this coming from?
Okay, great. Can you give us maybe, are there some risk factors linked to these adverse health outcomes? Like you did mention, maybe perinatal outcomes when it comes to artificial reproductive technologies, and are they mitigating solutions?
So from big population studies on children born after fertility treatments, the most obvious risk factors for this lower birth weight, which is considered a proxy of later on having other health issues, is so the main risk factors for this is first having a mother that is subfertile. So a woman that takes a long time to get pregnant. So the infertility itself seems to be a risk factor. And it's particularly for maternal subfertility. It's not paternal, but it's a maternal factor. And then there seems to be that any reproductive medicine intervention that involves ovarian stimulation also increases the risk. So the ovarian stimulation is a hormonal treatment by which you increase the number of eggs that are ovulated in one cycle, and this with the idea of having many eggs for your treatment. So any intervention, whether it's an insemination or an IVF or ICSI, it doesn't matter which one, but as long as you had the stimulation happening, this increases the risk of a lower birth weight.
Yeah. So to understand this better, your group, you and your team recently published a paper in Nature Communications, which is titled "Children Born After Assisted Reproduction More Commonly Carry Mitochondrial Genotype Associated with Low Birth Weight." What inspired this research idea, and what were your primary objectives?
Okay, so what inspired the research idea is a bit of a convoluted story, but let's say it was an educated guess. Like the short version is it was an educated guess. I had been doing other work on mitochondrial genetics in embryonic stem cells, and I had found aberrations in the stem cells, and I was wondering where they were coming from. Whether these had been inherited by the embryos that we use to make the embryonic stem cells. And so these embryos come from fertility treatments. And this basically started me pulling on a string of what would this mean if these embryos would carry these abnormalities. Without me knowing whether they would carry them or not. I was just fantasizing about this. I was just thinking like, okay, what would this mean? And I found it an interesting concept. And then I figured that this question about so I was aware of the question, why do these children have a lower birth weight? Because of the environment I'm working in. And so somehow the pieces started falling together. There was a lot of evidence for mitochondrial genetics and mitochondrial disease that suggested that people with mitochondrial variants would be potentially having a lower birth weight. So there were connections in there. So that's how I got to the hypothesis that the effect that we saw on the children could be caused by mitochondrial DNA variants. And then I built a whole model about how this actually could come to be. But a lot of people had been doing research on epigenetic changes. They had been looking at if these children had different epigenetic landscapes, but they don't. They have variability, but they couldn't pinpoint this to birth weight, right? So I was basically trying to solve the question on why these kids were different based on some hunch that I had that started at the stem cell research.
And I think it's important to say that this is the first of its kind to look
Yeah,
at low birth weight from a genetic point of view.
yeah.
Um, uh, because we always want to go from the basics. Somebody will be asking, what is the? We would like to know, what is the mitochondria? What is its role in fertility?
Yeah, well, mitochondria, I think whenever I say this in a lecture or anywhere, everybody finishes the sentence for me. Mitochondria are the powerhouse of the cell, right?
Powerhouse. Yeah.
So mitochondria are these out of endosymbiotic origin organelles that carry their own DNA, and they do many more things than making ATP. They are actually in charge of regulating apoptosis as well. They're making heme, their calcium metabolism. So they play many roles inside of the cell. And throughout development will play a baseline role in the development of the embryo and in the further health of the individual. So it's not that they especially play a role in fertility. They play a role everywhere. So anywhere that you have a problem with your mitochondria, you will have a health issue. The nice thing about the mitochondria in the context of disease or in the context of phenotype is that because mitochondrial DNA is a multi-copy gene, it's not a single copy like our nuclear DNA, you have something. If you want the nuclear DNA, if you have a mutation, you're either homozygous, heterozygous, right? So
Yeah,
it's an on and off switch, right? It's a very binary state of affairs. The mitochondrial DNA, it's a multi-copy gene, so you have a volume button. You can have 1% mutant, 10% mutant, 80% mutant. And for classic mitochondrial disease, we know you need to have a relatively high burden of mutant copies, right? But it's getting more and more clear that these copies that are mutations at 20% or at 10% or even compounding effect between different mutations can have a phenotypic effect and just give you ever slightly not so good working mitochondria, which then will give you a slightly worse outcome or different phenotype, you know. So that's the nice thing about mitochondria. So they have this yeah, it's not this black or white. It can be just a bit worse. And that was why it was making such a good model for the birth weight story, because children born after fertility treatment, it's not that they have a major problem. They're
yeah,
just slightly different. And that was, why I thought it really would work out.
yeah, indeed. So, can you summarize what are some of the mitochondrial DNA variants that you discovered in your research between children that are conceived through artificial reproductive technologies and those that are spontaneously conceived?
Yeah. So essentially what we found is that so we found two things and they're
Yeah,
connected.
yeah.
So the first thing we found is that anybody, whether you were spontaneously conceived or an ART child, anybody born with a lower birth weight tended to have more mutations in the mitochondrial DNA. This was irrespective of mode of conception. So this is a universal rule. If you're
Yeah.
born with these variants, you have a higher chance of being born with a lower birth weight. So these variants, specifically the ones that are potentially more deleterious. So we're talking about non-synonymous protein coding variants.
Yeah.
So any non-synonymous change in any of these proteins that form the electron transport chain in the mitochondria, and also variants in the rRNAs of the mitochondria. So the mitochondria have their own ribosomal RNA genes, and variants in these genes also cluster together with the risk of lower birth weight. So suggesting that these variants are giving you a slightly worse functioning mitochondria. And that was the model. So that's one thing. Second thing we found is that children born after fertility treatment, they had a higher incidence of non-synonymous variants. And also we went further and looked into eggs from stimulated and non-stimulated cycles and paired this with also children born out of the fertility treatments and spontaneously. And so that the de novo rate, and we could measure de novo because we had the mother and we had information of the mother and the child or of the donor and the oocyte. So we could see that these variants, they're not inherited, they're de novo appearing. So something during the procedure of ART was opening the door to having more of these variants. And in fact, what we found was that when you do a variant stimulation,
Yeah.
what happens is that you make a lot of eggs. You let the woman ovulate many eggs.
Yeah.
Now, this is not a normal circumstance. The normal circumstance is that you will ovulate 1 or maybe 2 eggs, right? So what happens in a natural cycle, in a normal ovarian cycle, is that there is a competition. There's like 10 eggs starting to mature, and they start competing for the hormones that they need for the maturation. There's a limited amount of hormones, and so FSH, and the eggs start competing. And the winner basically is the dominant egg and is the one that will ovulate. And in this natural process, you basically prune out eggs that are not as fit as the other ones. It's a natural selection process. Right? So when you do an ovarian stimulation, you obliterate this whole process. You let everybody pass through. All the eggs get ovulated. And also the ones that were just metabolically maybe not as fit as their neighbors. You see? And so essentially by having this ovarian stimulation, you obliterate a bottleneck, a selection bottleneck in which mitochondria so eggs with mitochondria that are not functioning that well, don't get selected anymore. Yeah. And so that's why after the treatment, you get more of these eggs with this variant.
Indeed. Now that is very elaborate indeed.
Yes, so, it is right, quite convoluted, I know.
Yes. So one thing that it's I think, because your study is actually bringing to light so many things that people have been questioning, or people understood that, okay, maternal age, maybe the cultural conditions, as well as the stimulation, like you just explained, also contribute to this low birth weight. But then there has not been an agreed standardized, maybe something like cultural medium to be used. And in this your study, you actually could really confirm the effect of oocyte cultural conditions for different cultural medium. Can you elaborate a bit on this?
Yes, actually, when you mention it like this, there's so many layers to the story, right? So indeed, the culture conditions were a confounding factor in the story. So we figured that in our group of children born after fertility treatment, we had been using 4 different culture systems for the embryo. Now, these culture systems, they are a black box. So we don't know what's in them. This is the company makes the medium, and you don't know what's in it. One could question whether this is okay or not, because so they clearly have an effect on birth weight. And there's quite a bunch of studies out there showing that different culture systems have different effects on the outcomes of the embryos. And so in our study, again, we see that there is a compounding effect, where with some culture systems, the children with mitochondrial DNA variants seem to follow the normal same trend as we find in spontaneously conceived. And with some culture systems, which are known actually to have the strongest effect on birth weight, they obliterate any genetic effect. So the culture system is so dominant that the genetic background, which is just a soft modulator, gets overruled by the culture system.
Do you think there will be some form of standardization on which culture medium to actually use in clinics? Is there any hope?
I'm afraid that here the interests of the pharma and the companies that are making them are very strong, and it would be very hard to push onto this. You would require a very large study that nobody wants to fund to be able to really say, okay, listen, with everything that we know, this culture system actually is the safest and best to use in a fertility treatment.
Yeah. So sometimes things like maternal age, smoking during pregnancy, pregnancy-induced hypertension, diabetes, are factors that normally affect birth weight.
Yes.
These are potential confounders in your studies. How do you differentiate these confounders to the mitochondrial DNA variant genotype association to low birth weight?
Yes, of course, we factored them in regression models. So indeed, hypertension is a very strong modulator of birth weight because you get early birth, and it has a very big impact on the fetus. Smoking, indeed, gestational diabetes, they've all been included in regression models. So we carry out either binary or regression, logistic regressions. We also had discriminant analysis models in which basically factor in these aspects that we know actually impact the birth weight.
Did you see maybe an interaction between the genotype, the variations, and some of these factors in predicting low birth weight?
That would be very interesting. I did look into it, but for that our sample size was too small.
Okay.
So at the end, I think to be able to do that, we'd need way more children with a lower birth weight. The problem is that because we were choosing deliberately people that had a birth weight under the 10th percentile, it means that only 10% of our individuals were in this range. So you need a lot of samples to be able to achieve that. And it wasn't designed really for looking into that, but it would be very interesting.
Yeah. In this study also you did show that children born with fertility treatment frequently show de novo synonymous variants than spontaneously conceived children. What might be the underlying reason for this increase between fertility treatment and spontaneous conceived children?
So the higher rate of de novo, we pinpointed the ovarian stimulation as the culprit, and not so much the fact of having the ovarian stimulation, but how strong the stimulation was. So the more eggs you retrieve, the higher the chances that you get eggs with these de novo mutations. Essentially, what happens is that since women are born with their eggs and they're sitting in the ovary, the mitochondrial DNA keeps on replicating, so it does acquire mutations, and so you have these de novo mutations sitting in the eggs there. And so if you have a very strong stimulation, you let pass a lot of eggs, you just simply have a higher chance of picking up some of these eggs with deleterious variants.
Okay, so when I went through the paper, I had this. I was reflecting on this question. So, normally mitochondrial DNA is maternally inherited.
Yes.
Can one say that women born through fertility treatment have an increased risk of subfertility and even worse for their own children due to the accumulated mutations from inherited and maybe precipitated by de novo mutations?
Maybe so. So this is we're just at the beginning of this story in the sense that people that are born from fertility treatments just now, the larger the larger cohorts are just becoming adults and having children themselves now. So we still largely don't know what the long-term effects could be of this. So there are some studies on male infertility, specifically from people from our group, that have shown that children born from fathers with severe male infertility actually also tend to have poorer sperm qualities. So suggesting that there is a genetic factor to that.
For sure. So this results, at least it brings to light so many things. What are some of the potential implications of your findings to the practice of fertility treatment? Are there any changes or precautions you would recommend?
So I think that the main thing I picked up from social media, from colleagues picking up on the paper, was that they were all taking home the idea of not stimulating the women too heavily. And I think that is what I would also advocate for, trying to find a sweet spot of having enough eggs to have a successful treatment and not exaggerating it, and not getting a cohort so large that the risk of picking up the mutant eggs gets too high. So it's really finding the Goldilocks zone in how many eggs do you need to have a good treatment and how much is risky. So we just submitted a follow-up paper in which we validated the findings on the eggs. So I think we're now pretty sure about the claim. It's an independent new cohort of eggs to show this. And there, again, I think the sample size is, we got caught in the middle of COVID while recruiting.
Yeah,
So
it was terrible time.
yeah, so our sample size is still not what I would have liked it to be, but okay. But it validates the first findings, and there I make some graphs to illustrate the relationship between how many eggs, and also the aging of the woman also plays a role in this. So trying to find an ideal area of how many eggs you should be collecting.
Yeah, so when I was looking at the outcome, I was looking from the point of view of a researcher and the point of view of maybe a patient or a client. How might this outcome influence, do you think, should influence maybe patient counseling prior to a fertility treatment?
I think patients should be always made aware that these risks that we have been observing for so many years, that they're there. And I'm not sure every center does this as thoroughly as they should, because people are very motivated to have a child. The wish to be a parent is extremely strong, and they tend, at that moment, prospective parents, they tend to minimize the risks in their heads. Even they're not able to, they don't want to see the risk, so they're sounding very minimal. But the risk of low birth weight is more than twice as high if you do ART than if you don't. Just in absolute numbers, yeah, sure. Instead of having a 10%, you will have a 20% chance. It still doesn't sound like a big problem, right? So it's very difficult, I think, to counsel people correctly in this. That I think invariably people will choose to just have a baby. And
Yeah.
it will be up to the doctors, to the gynecologists and the embryologist, to make sure that the treatment is done to the best possible standard. I think for the patients, I'm putting myself in their shoes, and I think it's very difficult. You're not going to say, well, then I'm not going to do IVF because there might be a risk that my child is born with a lower birth weight, which might associate later with cardiometabolic disease. There are too many hypotheses there, while the child wish is so strong.
Yeah, so in science, each time we find something new, it kind of shift our understanding of maybe the whole field. So what the question is, what is based on your findings, how do these results contribute to our broader understanding of genetics, reproduction, and child development as a whole?
I think something that is very nice, I think personally, is this generalized effect of the mitochondrial variants on birth weight. So it's not just the ARTs. The children after fertility treatment, they have a higher risk because of having a higher risk of getting the variants, but this applies to everybody. So I think that it opens the door to researching really how mild genetic changes can have mild effects. I think that that's something I'm very interested in. Is I think we in genetics we have mapped all the genetic diseases. We like the real bad things. We know them. All the Mendelian disease is mapped. We know it. What we don't understand very well is how our genetic blueprint is modulating mild things. Why some people are healthier than others. Why some people, I don't know, run faster or have a better athletic performance, or some people seem to smoke their whole life and don't get a cancer, and
And
others
nothing,
they
yes,
drop
those are
dead.
the questions.
So I think there is this whole world of mild phenotypic changes that's there to be explored, and I think this is one of them. This is how mitochondrial genetics impacts the onset of life, but probably also these same variants that were there at birth, they might be responsible later for instance, a higher tendency to cardiometabolic disease, maybe a higher risk for diabetes, this type of things.
That's quite interesting. So you also have another project that is currently running. It's on the genetic components to infertility and early development failure. Is this a continuation of this project, or can you give us throw some light on this project?
Yeah, this is a very large well, a grant, I would say. It's a fund from the university, and they are supporting the whole research line as such. So inside of this big name, like big grant, there are many projects running. So the mitochondria and infertility are still there. So there is a slow continuation of the project. So we're now looking more into doing retrospective studies on the children that are born from our IVF center. And so these are larger cohorts, and we're going to now, with what we know, we're going to try to see what effect the maternal age, the number of oocytes retrieved, and the culture medium have compounding onto each other on the birth weights. Without really looking at the mechanistic side, so just looking at how these factors actually, interact onto birth weight. We're also researching, where we're just starting now, looking for genetic factors that could explain very severe phenotypes of infertility. So couples that come and they never manage to get their oocytes fertilized. So they are injected, but they never manage. Then they never progress, or they always arrest at the third day of development. So really trying to understand that, to discover new genes actually regulating these very early developmental processes.
Yeah. Could you do you think maybe the long-term effect of fertility treatment on children, do you think it's something that it's worth investigating, or do you have an idea if there are other researchers looking into this already?
Yeah, I think this is still an ongoing topic of research. So in our hospital, there is Florence Belva, who is also the person with whom I work now for this study on the children. But the Scandinavian countries are very good at this. They have very large registries, and they keep on following up the children born after fertility treatment. So this is something that is still ongoing now.
Yeah, great. Could you discuss any collaborative efforts or support systems that were very crucial in the success of this research?
So this specific one, I worked together with the University of Maastricht. They provided about half of the samples that were included in the study. So it's not easy to recruit and get DNA samples from people, especially if you're are going to do genetic studies on them. So this was they were fundamental in the study. Without them, it would not have been possible. And then the people from the hospital, the university hospital at the UZ Brussels. The IVF clinic was incredible when it came to the study of oocytes. So there, we're working with very specific in this paper. We had oocytes from oocyte donors. So these are women that come to donate their eggs, and they volunteered to come. So normally they do these donations for other couples. So these women also came before their donation to couples to donate their eggs in spontaneous, normal, untreated cycle, so in a natural cycle. So this was amazing. And the people from the hospital, they did all the work to recruit them, to do the pickups. These women volunteered for this. So this was obviously invaluable. Without this, the studies would not have existed. Yeah.
So what do you hope to see change or improve in the field of assisted reproduction as a result of maybe your ongoing output bringing to light results like this one?
I think for me, what would be really important is that there is more evidence-based treatments in fertility treatment. So it's a domain that here in Belgium, it's very regulated, because fertility clinics are associated to university hospitals. It's regulated by the state. But in countries like Italy or Spain or the United States, this is a largely private endeavor. So it's private clinics. And so there is very few hold onto what exactly they're doing, what they're charging the patients for. They're selling all kind of additional treatments that are supposed to improve the outcomes, and all kind of things that are very monetary driven and with very few evidence. And I think if there's one thing I would like to see changing in this domain is this. I think that the model we have in Belgium, with the state reimbursed treatments is amazing, and also helps to put in a framework in which every intervention has to be evidence-based and not just something that would make money.
Yes. So, if what career advice would you give to young Claudia? Maybe during her bachelor's, maybe during she's still doing her bachelor's. In terms of what career advice would you give to her? She's very curious and very interested in a research career in science, in scientific research, to be specific.
I think I don't need to give myself any advice. I did exactly what I wanted to do, right?
Hypothetically, I mean young people who are interested in research.
Yeah, I think it's the same advice as I give my children and I give anybody, irrespective of what you're doing, right? There is, for me, two things to work. One is whatever you do it with passion and do it right, do it well. Whenever you do your job well, it eventually always gets recognized. I think that this is key. It's fundamental. Is do your thing, do it well. Take pride of what you're doing. It doesn't matter what it is. And then the second is follow your dreams to a certain degree. So don't be afraid of taking risks and following your gut, because that's what is going to bring you to that job that you are going to be doing right. So I think that's more the type of advice I would give.
Professor Speed, thank you very much for your our insightful discussion. And I think we come to the end of our discussion and interview of today. To our listeners, thank you for watching, and stay tuned to the next edition of our discussion. Bye.
No, thank you.
Yeah.